to go over what is a mammal, what defines the clade mammalia, and also what do I mean when I say clade mammalia. Seems like an easy question. What is a mammal? I'm sure you have an idea of what is a mammal in your head already. You're probably like, well, fur and lactation and gives birth to live young. Well, especially each one of those, you might say, oh, well, what about whales? Do whales have hair? Oh, what about gives birth to live young? Aren't platypus and echidnas, aren't those mammals? And they don't do that. Do they lactate? What actually is and isn't a mammal? And kind of the best way to start thinking about this is by thinking, what isn't a mammal? Here's one of those not mammals right now. It is generic lizard. This isn't a mammal. So why do you immediately look at this and know this isn't a mammal? Well, I'm sure things like fur and a bunch of other things are coming to your mind right now. So here is a mammal. This is a shrew, some variety of Sorex, which is a very good representation of a primitive mammal. So before you go to the next slide, take a few moments and just think, what are the things, the morphologies that you see as the most important differences between the non-mammal and the mammal? Okay, so what are the things that you most notice that separate this lizard from the shrew? And fur, another thing, ear. There's not much of an external ear on the shrew. It's right here, but notice we don't have this big hole. You have ear flaps on the side of your head, not big open holes. This is the reptile ear. Also, think about the position of this ear relative to the jaw. What else? Well, the mobile snout, the size of the eye, perhaps, the orientation of the legs. All these and more are things that immediately separate mammals from non-mammals. So before we go into more detail, we're going to go into explicit, detailed anatomical differences. We're going to look at some trees and say, okay, in the scheme of things, what is a mammal? Oh, what are mammals? Mammals are deuterostomes. So if you don't remember from intro biology, we can divide the bilateria into two main clades, protostomata and deuterostomata. Deuterostomes are the clade that includes our phyla, Chordata, as well as the phyla that includes starfish and their relatives, Echinodermata. And the things that group these animals together are very early embryology, such as the fate of the blastopore, how cleavage occurs, when differentiation of cells occurs, and onward. Mammals are deuterostomes. Mammals are chordata. They're chordates. And inside chordates, you have, a notochord, it's in the name, as well as a series of other features, such as a postanal tail, segmentation, dorsal hollow nerve cord, pharyngeal slits. We have those at some stage in our development. Not the whole time, but some stage they're in there. What else are mammals? Well, mammals are craniates. There is some sort of a formed brain and a cranium. Mammals are vertebrates. We have a clear skeleton, including a backbone, a vertebral column. Mammals are gnathostomes. What is that? These are animals that have jaws. And then farther up, mammals are, osteichthyans. We are actually bony fish, the clade that includes things like goldfish and perch and all that also, includes the lobe-fin fishes, which are our closer relatives. Farther in there, mammals are tetrapods. Mammals are vertebrates that have 4 limbs that, at least primitively, support weight. And with inside tetrapoda, there are only 3 main groups, amphibia, reptilia, and mammalia. Here we are. And mammalia, together with reptilia, are amniota. These are the amniotes. So this, in the overall tree of life, this is say what mammals are. And see here, mammals are, one of the big things that separates them is production of milk. Can produce food for their young. If we look inside, amniota more, that's right here, you see that, there's quite a lot of diversity within inside reptilia, and that includes birds. Birds are a member of reptilia. using cladistic terminology. And then we have mammals. And as we'll see when we go through the mammalian family tree, a lot of the forms that kind of connect mammals more closely to what we consider reptiles are no longer extant. Monotremes are the most primitive living mammal, and they are quite different. But there were a whole host of other forms that, if you saw them just walking down the street, you would think they were a reptile that trace back this common ancestry of reptiles and mammals. So to get, this is a clade. Reptilia plus mammals makes amniota. We can say reptiles are the sister clade to mammals. And, this is just another way of looking at it. It's kind of these extinct groups that we have. So this is the same tree as before for the most part. See, we have amphibians here, and here we have the reptiles that here are called Sauropsida. It's kind of a more scientific name, but you can think Sauropsida basically means reptilia, kind of. And then, we have Synapsida. Synapsida is a group that is mammals. And so here, we have dinosaurs and birds, which are living dinosaurs, crocodiles, things you may have not heard of before, some extinct clades, mesosaurs, extinct aquatic mammals, not mammals, extinct aquatic reptiles for the most part, and then we have synapsids. So we just have mammals here, then we have all these extinct groups that kind of connect us to where, at the base of amniota, the mammalian line diverge from the reptilian line. And we'll go through a lot of these groups in the first few lectures. Okay, we got reptiles over here, mammals over here, and they look very different. But if we go back to their last common ancestor, they looked very similar, and mammals have acquired a host of features that separate them from the modern reptiles. And if we go numbers, how many mammals are there? We're actually losing in terms of raw numbers. There's just over, well, maybe just under 5,500 mammals. There are 10,000 birds. There's 10,000 reptiles, not including birds. So, there's 20,000 reptiles altogether. And there's even more amphibians than mammals. And most of those are frogs, not all of them. There's also salamanders and limnous amphibians called caecilians. But mammals, as much as we think of mammals as the dominant form of life on the planet today, you'll see a lot of stuff like, oh, the Cenozoic was the dawn of mammals, and mammals have dominated, we're mammals, so we like to think we're the best. Numerically, we're not doing that great. We are the least speciose tetrapod group. And if you get back in the water, if we're not just looking at tetrapods, there are way more fish, the fishy fish, non-tetrapod fish, than any of these groups of tetrapods. How does it shake out inside Mammalia? So we'll get rid of all those other tetrapods, and what do we have species-wise? Well, the majority of species of living mammals are placentals. These are placental mammals. You are a placental mammal. There's over 5,000 species. The other groups are marsupials, marsupialia, and monotremata, the monotremes. And how are they related to each other? Fairly simple tree here. Marsupials and placentals are each other's closest relative. These are a sister clade relationship, and together they make the group Theria. Outside Theria, we have monotremes. And this probably isn't surprising if you think about it for a little bit, considering, both marsupials and placentals give birth to live young. It's very rudimentary, very early stage in marsupials, but it's still live young, whereas monotremes lay eggs. And there's a lot of evidence, both morphological and molecular, that supports this grouping where marsupials are more closely related to placentals than either one is to monotremes. Together, this is Mammalia, this is clade, and then we have Theria. So when we say mammal, we mean any living mammal, including monotremes. If you want to just talk about marsupials and placentals, the mammals that give birth to live young, that clade is Theria. So we see right here, to be a mammal, you don't have to give birth to live young. So what are these defining features? So we're going to start to go through the defining features of mammals. And we are going to talk about some features that monotremes don't have. And so this is one thing where you don't necessarily need all of these features to be a mammal, but if you have these features, you are definitely a mammal. Let's start with the feature that has given the clade its name, mammalia. That name is based on mammary glands, the milk secreting glands that mammals have to feed their young. And mammals are the only animal, is the only vertebrate animal, we're going to leave insects aside because they do everything, the only vertebrate that can produce a nutritious substance for their young that is not just regurgitating food that it's already eaten. So you may say, like, oh, well, I thought, like, pigeons can kind of feed their young something for their crop. Well, they can, but what that is that is just food they have eaten and stored, and they'll give to the young later. It's not actually producing new it's not actually producing a new substance like mammals do. So, here we have a pig, we have a manatee, we have a rat. All of these mammals are nursing their young, giving them milk that they are secreting from glands. And primitively, mammals have an entire row of these glands, such as in the pig and in the rat here, but several species have restricted these glands to only be in certain places, such as manatee, they're only just underneath the arms. Maybe a cow just has that one udder. And so what here's a cow right here. What are these? So here is some mammal trivia. This is a nipple, which is very closely connected to the milk ducts and the glands that secrete the milk. Whereas what you have in what cows have, we have this teat, where you have these glands are much deeper relative, like they're not as superficial, and you have the milk collects in the cistern, and it is expressed out of this teat here by the young, or farmer milking the cow. And monotremes do secrete milk. However, they don't have nipples or teats, either one of those. This monotreme is feeding her babies, which are called puggles, by the way, her little baby puggles, via just milk that is almost sweated out of her body. So she has these row of glands on her abdomen that secrete out milk, but it's more like sweat, kind of like milk sweat is a lovely term right there. And so the delivery mechanism is not very refined in monotremes, but it's thought, this is what the more developed nipples came from in later mammals. So milk is something that all mammals do have, including the monotreme. What other glands do mammals have? Mammals actually we're really glandular compared to reptiles. Reptile skin is always really dry. The scales are really, really thick. Not much is getting in or out. By comparison, our skin is much, much thinner, and we use it for a lot of waste disposal and for thermoregulation. Not something reptiles really have to care about too much. So it's not just that, we have hair coming out of our skin, but we have sweat glands inside, we have sebaceous glands, and we have musk glands. Think about any time you've seen a dog or a cat or sugar gliders who rub the side of their face against anything, they're giving their scent there. That is a very mammalian feature, that musk gland. It is really common. It's not just dogs and cats. Deer have it as well. Many, many mammals have some sort of gland that leaves their scent, that kind of marks their territory or is used for communication. Again, hair. Hair is a mammal feature. And what is hair? Well, hair is, as it comes out, it's not alive anymore. It's only alive at the root. And it is a very tightly compressed keratin sheath, so to speak. It has pigment granules inside, maybe. It's got a core, which is called the medulla, and it's got a cortex. And the shape of hair, like, is hair soft, is hair curly, is hair straight? A lot of that has to do with how the keratin on the cortex is formed. And hair is, of course, mammals have it to keep themselves warm in cool environments because we create our own heat. But mammals also have hair in very warm environments. So, hair is not just an insulating thing. Hair is also protection. How is it going to protect you in the desert? Well, the hair around the eyes can keep sand from blowing into the eyes, but also, I don't think this fennec fox has to put on sunscreen. Hair also protects you from the rays of the sun. So, hair is not just insulated. Fur is just really short, dense, fine hair. Not always that fine, but hair and fur are the same thing. Hair is not only an insulator, but it's a general protective structure. And it's not just protective in terms of protecting the body from the elements, but it can protect you from other things. The pattern that baby deer have, the speckling, like this actually helps them hide from predators. If you've ever been going through the forest and you just find a deer laying in the shrub like this, it's not going to move because the spots on here that it loses when it becomes an adult, they protect it. They break up its outline from predators. You might be like, but I can see that clear as day. We have really good vision. A lot of natural predators for deer would not be able to see this deer very easily. And here the masking that a raccoon has, this actually serves a couple features. It helps to kind of like the same way a football player or something puts the markings under the eye to kind of help with playing it being in the sun or all that. But it also serves as a kind of mask to make it so the eyes look bigger. So especially at night, you may not be sure where the animal's eyes are or how big they are. So the coloration of fur is really important for a lot of mammals' protection as well in day-to-day activities. What about default coloration? There are some animals that have fur, many mammals that have fur that don't necessarily have a special pattern. And what we have here, exemplified by this deer mouse and this mouse deer, that's a mammalogy joke, big hit at parties. This is kind of the default mammal coloring, where the dorsum, or the back, is a dark color, and the ventral surface, the underbelly, is a light color. And this is kind of the default coloration you're going to see in many, many mammals over and over and over again. And why is this? Well, first of all, if you are high above these animals looking down, say you're an eagle looking for a tasty dinner, and yeah, these are taken by eagles sometime, this helps you blend into the surroundings more. So you might be like, well, why not be brown all the way around? Well, if you are looking from the side, this kind of will break up the outline of the animal, and it will make it so that it's not as easy to see, because that the idea, like this might be at the horizon line where the white part kind of blends in more with the ground, and the brown part kind of more the sky or a branch behind it or a tree or what have you. So this is kind of the standard mammalian pattern. So if you see something like this, there's not going to be a lot of people researching that, like, well, why do they have this? Because this is primitive. This is the default. When you see mammals that have a different coloration, such as stripes on a tiger, stripes on a zebra, stripes on some other thing, that is when you want to kind of ask questions. So like, why do we have this coloration? What is it doing? And sometimes the answer is really simple, which, Arctic fox, why is it all white? Because it lives in the Arctic, and being brown on the top isn't going to help it out very much. But there is a reason, typically an evolutionary reason, that you can explain a deviation away of why clade acquired a different coat color than these primitive ones. Now you might be like, do all mammals have fur? And the answer is yes. So here we have a pangolin. This is a mammal, not a reptile. Pangolin, and actually it's a really derived mammal. It's not very primitive. And this is sometimes called the pinecone mammal, or the scaly anteater, and it does have fur. Now, these plates, these are keratin. They're made of the same material as your fingernails, and you may remember, I said hair is keratin as well, but these plates are not the homologous to fur. If you zoom in here and you look at all the scales of a pangolin, there's actually still very coarse fur that comes from between the scales, and its underbelly is covered with this fur that has a straw-like consistency. So we see fur even in mammals that don't first appear to have fur. And even a whale will have some whiskers, and we'll talk about this more when we talk about evolution of mammals, but it's thought that whiskers came first before the hair on the rest of the body. The vibrissae, the hairs on the muzzle, the snout, those probably came first. And you can see those in a lot of animals that don't have hair on any other part of their body. Another thing mammals do, store fat. So, we have blubber here in the seal, or this very, very successful prairie dog. Don't worry, this was taken in a zoo. It doesn't have to worry about running from predators or even fitting into its burrow. And this you may not have thought about before, but this is a very mammal thing. I mean, we don't really see, fat lizards. We don't typically see fat birds, though it can happen. It's not nearly as common as you see fat mammals. Mammals are really, really good at storing fat. And when we do see non-mammals that store fat, such as this gecko, it's typically one part of the body and not throughout the entire body. This gecko, has this really fat tail because this is where it's storing its fat. And, if need be, if it needs to run away, it can drop this tail and then, so it can have the benefit of having a bunch of reserve fat for if times get hard, but not necessarily be encumbered by it if it's going to greatly put it in peril. Another thing that separates mammals from most reptiles, and I say most because birds have the same thing, is we have a fully four-chambered heart. If you've taken anatomy classes, you've heard, the right atrium, the left atrium, the right ventricle, the left ventricle. What you may not know is that that is a special mammal or bird thing. If we look at most reptiles, for example, they don't have a fully separated ventricles. There actually is some interplay between the left and right ventricle. Amphibians have a completely they only have one ventricle. They have a middle ventricle, they have a left atrium and a right atrium, but just one. And you look at fish, they just have a single ventricle and a single atrium. So we have developed a four-chamber heart over evolutionary history because this is more efficient. You have no blood mixing possibility in this. Another thing that mammals have that's very different from all other animals, and now this includes birds, birds don't have the mammal condition, is we do not have nucleated blood cells. Our blood cells, our red blood cells, do not have a nucleus. So, they can't divide. They have to come from our bone marrow. Most other vertebrates have a nucleus in their red blood cells. Now, even though all mammals have a very similar heart, right ventricle, right atrium, left atrium, they don't all have the same rate, and this is something that probably makes a lot of sense. You wouldn't expect an elephant to have the same heart rate as a shrew or a rat. And you can see here at this table, a shrew heart beats an incredible amount of time, 782 beats a minute, and there's a huge range on that. A shrew heart, if you were listening to it, you couldn't even make out the individual beats, whether if you were looking at a Phoca vitulina, that's a seal, type of seal, a harbor seal, says right there, you only have 18 to 25 beats a minute. You might be wondering, is my stethoscope working before another beat came along? Moving from the heart to the lungs, mammals are very different here as well from reptiles. As you might be familiar with, your lungs, you have your trachea, your common trachea, that then divides into many, many tubes within your lungs, and they terminate after many divisions into the alveoli, where interface with the capillary system, your capillary part of your circulatory system, and your blood is oxygenated, and carbon dioxide goes out, and here comes the oxygen. It's very, very regimented. Not so in reptiles. Reptiles, instead of having clear pathways, kind of like a highway where the air goes, instead their entire lung is kind of a homogeneous sponge, and air goes in and kind of diffuses throughout this lung, and they push the air out with action from the muscles, very similar to what we do, creating a vacuum and then contracting that vacuum. But it's not nearly as efficient. But it doesn't have to be because their metabolism isn't as efficient. And this is an ornithology class, but birds do have a lung like the typical reptile lung, but they have a completely different way of interfacing with the circulatory system, so they maintain their higher metabolic rate a completely different way than mammals do. But the mammalian lung, which a lot of people tend to think of like the standard lung, is highly specialized in our clade. Of course, the mammalian reproductive system is one of the defining characteristics of mammals. There we have live birth in most of them, not quite the platypus. It does lay eggs, but in mammals that have live birth, there is the uterus where the young are held inside the body, and especially there is a placenta. And this is the thing that really separates mammals from everything else, because many clades do have live birth. There are snakes that give birth to live young, fish, sharks, some other lizards, not just snakes. It is actually really common. We never see it in birds, but we see it in many other groups. But those are just eggs retained inside the body. It's not the mammalian system where you give nutrients to the growing fetus via the placenta. And there is the scrotum as well. Here we have a male rat and a female rat, and you see the testicles here contained inside the scrotum of the male rat. You don't see a scrotum on lizards. If you want to determine the sex of a lizard, it's a little complicated. An alligator, things like that. You can't just look at them, flip them upside down, and figure out if you're looking at a male or a female. And sometimes it's coloration stuff, but not this external anatomy. Well, what is going on here? Why do mammals have this? Well, it's because of our higher body temperature. So lizards do have testicles, but they're in the abdomen. They're actually in about the same place that the ovaries are found in female mammals. And what happens is in our growth, the testicles start in the same place, and as the mammal grows, they descend down through the abdominal wall and fall into the scrotum. And this is because the mammalian body temperature gets so high inside our abdominal cavity that it would not produce viable spur. Mammals dispose their nitrogenous waste, creating urea, which is kind of a compromise way of disposing of nitrogenous waste. It takes a lot of water to not make it toxic, not as much as ammonia does. It just takes some energy to produce it, not as much energy as uric acid takes. Again, a lot of review from IntroBio. Mammals are not the only animals that produce urea. We also see it in some amphibians, some turtles, and actually turtles can do different things depending on their environment. And some the lobefin fish do this as well, and sharks. But what does separate mammals is the mammalian kidney. Mammals have perhaps arguably the most efficient kidney of all the tetrapods, with its different levels of concentration. So you can have osmosis as you have fluid go up and down the nephrons. The cortex is not nearly as dense as the medulla, and then all the many meters of nephrons that are in each kidney allow for really efficient filtration of the blood, and it brings out as much water as possible, so we keep a lot of water inside the body, and we eliminate just as much as we need to safely dispose of the urea. Brains. Mammalian brains are another hallmark of our group. So here's a little bit of brain anatomy. So here we have a human brain and alligator brain, and if we color everything about the same, we see that first of all, you can't really see the olfactory bulbs in the human brain. Well, that's because we have a very bad sense of smell. But you notice the cerebrum. Oh, here, I'll color that one green yet. The cerebrum is super large in the human brain. Well, how does this compare with other mammals? So, like, here's a squirrel. There's coloring. And you can see that even though it's not nearly as big as in the human brain, the squirrel has a quite large cerebrum as well. So it is a hallmark of our species to have extremely well-developed brains with gigantic cerebrums, but this is a mammal feature in general. Mammals have much more developed cerebrum, much more cortical folding typically than we see in non-mammals. So what is the big diagnostic feature of mammals is an increased cerebrum. And if you remember from other classes, the cerebrum is where you kind of have processing. It's where you you synthesize your visual, auditory, other information coming from your senses. It's where, you decide, I'm going to move this way, I'm going to move that way, I'm going to grab this, grab that. And the cerebrum is much larger in mammals than we see in non-mammals, even accounting for body size. And here, this is a kind of insectivorous brain. I believe this is a hedgehog brain. And our cerebrum is much larger than the alligator, but notice we don't have all these cortical foldings. This is something you see that as mammals get bigger and bigger, you start to see folding of the brain, and you even see it some of the small mammals that have quite great cognitive capabilities. So you increase cerebral size. Another thing that mammals typically are pretty good at, humans not so good at this, is the sense of smell. We have an extremely good sense of smell. I said the clade mammals. And the reason for this is that if you look at a reptile kind of brings the external nares in, brings the air in the external nares, and it goes to the nasal chamber, and then it kind of goes into the mouth. And you have some epithelial tissue here that, takes the molecules from there and has the sense of smell occurring. But what mammals do, you see here by these little lines here, if we look here at this dog nose, these bones here, these are the choanae of the nose, and these are called the turbinals. This is where tons and tons of epithelial tissue lies that greatly increases the surface area to capture the smells from the air, and so mammals have a very great sense of smell typically. We don't. We've lost our sense of smell. Primates in general have much better vision than a lot of other mammals and a really not great sense of smell, but primitively, mammals have a really good sense of smell, and the anatomical thing you see of this is when we look through the noses of the nasal aperture, use the fancy term, we'll look through the nasal aperture of the skulls in lab, you'll see this in pretty much all non-damaged mammalian skulls. Another, moving through the sensory organs, mammalian hearing, and we'll go through this in much greater detail when we look at the evolutionary history of mammalia, but mammals hear a very, very different way from non-mammals. So you might have heard about, your three middle ear bones. You have the hammer, anvil, and stirrup, or, the names here, we have the malleus, incus, and stapes. We have these three middle ear bones. Reptiles just have the stapes. We have additional middle ear bones that have actually come from the jaw joint. We have a very different jaw, and we'll see this in lab when we really look at the skulls. Mammals, we have one bone in our lower jaw. We have the dentary or the mandible or whatever you want to call it. Reptiles have their dentary and the angular and the articular and all these other things in the lower jaw. And our jaw joint is very simple. It's just the dentary articulating with a squamosal. You have a much more complicated jaw joint. So here we see in this primitive synapsid Dimetrodon. Dimetrodon is more closely related to you than it is to a dinosaur. It is not a dinosaur, and it's not a reptile. And so here we can see the ear of a squirrel to see how complicated the mammalian ear is we have this apparatus here, and we have the incus, stapes, and the malleus, where it greatly magnifies sound going to the ear, and it's been shown that mammals have a much higher pitch capability. That's one reason that very small mammals have high-pitched squeaks is how they communicate, because a lot of reptiles can't actually hear that at all. And if we move to the outside of the ear, looking at the soft tissue structures, and I'm using our deer mouse again instead of our shoe, is here we have the external pinna, this cup on the outside of the skull that captures sound and allows the mammal to hear much better than here. You can see this lizard just got a hole in the side of its head, no kind of satellite dish to capture the sound, and you can actually see the stapes is right here. The final soft tissue structure we're going to talk about that really serves to greatly separate mammals from reptiles is facial muscles. Feel your face right now. You have all these muscles there. Think about the expressions that you can make, and you have lips, you have cheeks that you can pout, put stuff in, or breathe out, or suck them in, or all this, squeeze your eyes shut, raise an eyebrow, all of these things, or in a lot of mammals, wiggle their ears. Maybe some of you could wiggle your ears. Lizards, which is a pretty standard reptile, no, they have almost no facial muscles. And that's actually why when we're going to see in lab, we compare reptile skulls to mammal skulls. A mammal skull looks very, very different than the living mammal does. A lot of the reptile skulls look pretty much the same, and it is because of the almost complete lack of facial muscles in reptiles. That brings us to the skeletal features that separate mammals from reptiles. And I'm not going to go into much detail in these, as we're going to investigate a lot of these for ourselves in lab, but I'm still going to go over them in the lecture. So one of the first things to look at as we go through some basic anatomy of a long bone, we have what we call here is these are the epiphyses. This is the end cap, you think what the joint is. The metaphyse is the interface between them, and this in mammals fuses together at the cessation of growth. Then you have the diaphysis or the shaft, and then on the other side you have another epiphyse and metaphyse. And this is not humerus, a femur from a hedgehog. And basic anatomy terms, proximal is closer to the body midline, distal is farther away. So your hand is distal to your elbow is proximal to your hand. As a general, we have some generic here, cow skeleton and crocodile skeleton. So you can take a moment just to look at the cow and the crocodile and think for yourself, what are the big things that look different between these two skeletons? Oh, here we have, I believe this is an Allosaurus, maybe a juvenile one, but some sort of theropod dinosaur, and we have a mammoth. And one thing you may have noticed when you look at these large mounts in museums is that dinosaur bones, notice they don't actually touch each other. So here we have the mammoth knee. Actually, no, I think this is just an elephant, this living elephant. I think it's mammoth, but it's not a fossil. But we have this elephant here. The knees do contact each other. They touch. We do have this joint here. We don't see that in the dinosaur, and it's not because, oh, this is a fossil and we don't want it's too hard on the skeleton if they touch each other. No. Reptiles don't ossify the epiphyse. What that means is that even when fully grown, there is a cartilaginous cap on the bone, and that is what contacts the other bone. They don't actually have fully ossified ends like we do in mammals. And mammals, you always have this ossified end, but what it is if you have a juvenile, we see this elephant wasn't quite an adult when it died, because you can clearly see the growth plate lines between the ends of the bone and the middle of the shaft. Now that's a big difference, where if you just found a random bone, how you can easily say, oh, this is a mammal versus this is a reptile, even if you don't know anything beyond that as to its attribution. So we're going to go over some detailed aspects of the cranium. So on the left, we have our representative reptile skull, and we'll have one of these to look at in lab. This is a tuatara, which is a very primitive reptile. It's alive today. It's not a lizard, but it's sister taxon to all living lizards, and it has a lot of primitive reptilian features on the skull. And on the right, we have a wolf skull, which is a pretty good representative modern mammal skull. Now, again, we're just going to have a whole host of things pop up here because we're going to go over this in more detail in laboratory, and I want you to investigate for yourself. But some of the big things that separate mammals from reptiles is our skulls are akinetic. What does that mean? There's no motion. We'll look at the reptile skulls, and there are many different joints that move in the reptile skull besides just the jaw going up and down. There's also only a single craniomandibular jaw joint in mammals. You can have kind of multiple ones in reptiles. Mammals have a much larger brain case. It takes up basically the entire back of the skull. This is the brain case in the sphenodon right here. It's very, very small. There are crests in the midline of the mammal skull that we do not see in reptiles. We have the zygomatic arch which goes on the outside, which has a muscle that runs underneath it. There's a secondary palate, so if you push your tongue against the roof of the mouth, you hit some bone there. If you were a reptile, you'd be able to push your tongue up and just kind of touch the inside of your nose. But there, as we talked before, there's terminal bones inside the nose. There's many foramen where the cranial nerves and vessels have to go through. In a mammal skull and a reptile skull, there's so many holes they just go whenever. Again, there are 3 middle ear bones in the mammal skull, just 1 in the reptile skull. There's this tympanic bulla. We'll again talk about that in lab. There's this enlarged bone that it has that you can see right here that creates this pocket of air inside the skull, which houses the middle ear bones. And there's a thing called the hyoid, and you can actually feel it if you feel your neck. There's a hyoid bone in the middle there, and that helps to support the tongue. It's much more developed in mammals than the homo in reptiles. And again, we'll go over these in lab in more detail. Overall, we can divide all vertebrate skulls into a couple of major categories. And here we can see in this figure I've taken from a text, we have 4 skulls. Up here in the upper left, this is called the anapsid skull, or kind of the primitive amniote condition, where the only hole in the lateral margin of the skull is the eye. So here would be the eye socket right here, and that's the only opening in the side of the skull. What we are, all mammals are a form of synapsid skull. We see here the hole where the eye goes, and posterior to that is a second opening. This is a synapsid skull. Reptiles are diapsids, where here is the opening where the eye is, and behind here are 2 holes for allowing muscle attachments. And here, D, this is called a eurapsid skull. This is a modified form of diapsid skull that we don't need to worry about for this class. This is for marine reptiles. So what are the purposes of these holes? So remember, anapsid, kind of the primitive condition, synapsid, that's us, diapsid, reptiles. Well, what these allow is here, if you can see, we have the lower jaw here, and here is the skull roof with a little brain cavity here, and this is the muscle attachment attaching the lower jaw to the skull. Well, if you have these openings here, or here the openings in the modern mammal, it allows you to build up bigger muscles, to have a bigger brain case and have bigger muscles so that you aren't sacrificing anything as your brain gets larger for bite force. Another trend we see overall going from the reptilian condition or the primitive amniote condition of mammals is a huge reduction in bones, such as in the lower jaw. Primitively, reptiles have multiple bones in the lower jaw. When we get to a modern mammal, such as us, we only have 1 in the dentary. We'll see this overall, a repeating pattern is that there are just many more bones in the reptile skull that we don't have in the mammal skull. And sometimes, even if we have the same amount, they're just much smaller. What about in the postcranium? What are some big changes in the postcranium from the reptile or the primitive condition to mammals? Well, the bones are mostly all there, all the same ones, but there's some gross differences. For example, our ribs don't go all the way back to our sacrum like we see in reptiles. They also have cervical vertebra. We don't have any ribs on our cervical bones. If we get in detail, there's a lot of transformations of the girdles. This is one thing. Think about how our reptile stands and how our mammal stands. We put our limbs underneath ourselves. We're very upright posture. Reptiles have sprawling posture. And actually, as we'll see in the platypus skeleton in lab, mammals started out very sprawling, and it took them a long time to stand up. And here, so we have the shoulder girdle of a representative mammal, here Erinaceus, and iguana. Much more complicated looking in the iguana. We've lost bones such as the inner clavicle and the coracoid going from reptile to mammal. Now we just have a process on the scapula that we call the coracoid process. So we've gone from many bones to 2 bones in the shoulder girdle that, of course, the humerus would go on here too, but these are the bones of the midline. Now moving to kind of the last big section of hard tissue anatomy to talk about is dentition. So we're going to be talking about mammalian dentition a lot. We're going to be talking about the evolution of mammalian teeth. But the thing to know about mammal teeth is what makes them special is we are thecodont, diphodont, and heterodont. What does any of that mean? So first of all, the dont term there means it has to do with dentition. So what we mean by thecodont is if we look at a lizard condition, the teeth are adhered to the side of the jawbone. They are not actually embedded in any bone. The mammalian condition, thecodont, is where the tooth is surrounded on all sides by bone. Now, this is not only a mammal thing. Some reptiles have actually developed this type of dentition convergently, such as alligators. But, if you look at a lizard jaw, it's going to be pleurodont. So we are thecodont. The teeth are embedded inside bone, both the lower jaw and the upper jaw. We are diphodont. What does that mean? 2 generations of teeth. And typically, there are exceptions, but only 2 generations. We had our baby teeth, also called the milk dentition or the deciduous dentition. And then we have the adult teeth that grow in after that. And, our teeth are divided into molars, premolars, canines, and incisors. And it's pretty easy for people to see what makes incisors incisors and canines canines, but I commonly have people ask me, like, what is the difference between premolars and molars? They can look very similar sometimes. And that is, they can. The difference is premolars have a deciduous precursor. You have baby versions of your premolars. Molars don't have anything before them. You didn't have baby wisdom teeth, for example. They just grew in. So if there's no precursor, it's a molar. If there is a precursor, it is premolar. And that actually already explains what we meant that last term. Heterodont is different teeth. We have all different types of teeth in our jaw. Like I just said, incisors, canines, premolars, and molars. A primitive way that vertebrates have teeth is just the same thing over and over. There's the same little peg nonstop. Now, how many molars and how many premolars and how many incisors varies tremendously from mammal to mammal. It's one of the things that identifies different species. Just as a quick example here, you can see this graphic on the right that a lot of cats, such as the lynx here, have very little molars, and instead of their meat processing is on the premolars. Primitive mammals, such as Canalestes here, did have a full set of molars. However, a lot of herbivores still have a full set of molars, and we have a fairly full set of molars. So when you're looking at a mammal and you're trying to identify the number of teeth, don't try to apply the human condition or even the primitive condition all the time, because there's a lot of variety out there. And there's so much variety that we've developed what's called dental formulas. What a dental formula is it's a quick and easy way for a researcher to instantly know how many teeth a mammal has in its mouth. And the dental formula here, it's specifying the number of precision teeth on one side. So think about dividing the mouth into 4 quadrants, upper right, upper left, lower right, lower left. We assume there's left and right symmetry because that is how things go, bilaterally symmetrical animals. That's one of the things mammals are. So if we have 3 incisors on the right side, we're going to have 3 incisors on the left. But we don't necessarily have the same number of teeth on the top and the bottom. You might have 3 incisors on the top and 2 on the bottom. And sometimes you only have upper canines or you only have lower canines. So we need to have a way to ascribe how many we have on both the top and the bottom. So see here, if we're talking about an animal that has 3 incisors on the top and bottom, 1 canine on the top and bottom, 4 premolars top and bottom, 2 molars on the top and 3 on the bottom, we would write this as such. 3, 3, 1, 1, 4, 4, 2, 3. And it's just, that is the standard that from left to the right, it's the number of incisors, canines, premolars, or molars. And individual teeth, if you're just talking about a single tooth, you're designated with an uppercase letter for upper teeth and lowercase letter for lower teeth. So if you're reading something and they're talking about the diagnostic feature of an animal is a hypertrophied, a very large capital P3, they mean the 3rd upper premolar. If it's a lowercase m2, they mean the 2nd lower molar. So to sum up, here, and this is from your textbook, these are what some of the main diagnostic characteristics of mammals. And there's a lot of soft tissue structures here, as well as hard tissue structures. I don't agree that these are necessarily the most informative ones. As you can see, this is selected from a list. But, these are a lot of the features that kind of hit things about the brain, the hard tissue, hearing, glands, kidneys, all these other things. So don't memorize this list, but just be aware there are a lot of differences because you will need to provide several of them. Now, we also see some trends, and what do I mean by that? Well, these are things that aren't necessarily rules, but they're kind of trends within mammals. So, one thing is that, mammals are endotherms. So if you have a mammal and a lizard about the same size, though, there's not going to be things that are equivalent. For example, lizards can be much smaller than mammals and still going to do their lizardy thing. So if you're comparing, you just think about it. These are things even if you haven't really thought about them in the scientific setting. Think about how a lizard runs on the campus versus how a shrew would run. First of all, you're not really going to see that shrew during the day. The lizard's going to be really long. The mammal's going to be really spherical. Mammals get really round pretty easily. And so, these are things just keep in mind is that a lot of the ways mammals act, mammals behave, is because we are endotherms instead of ectotherms. And again, to sum up, there are, probably about 5,500 species of mammals. Most of them are placental mammals, the vast majority. And then we have marsupials, which we have one marsupial currently alive in North America, the Virginia possum. They're mostly in South America and Australia, and monotremes are entirely found in Australia and New Zealand. But what if we want to talk about a specific mammal? For that, we need to go to what you're probably familiar with is, so how do other scientists communicate if we want to talk about a single species, such as this jackrabbit? Now, it might have many different names. It might be a common name as a bunny, or just a jackrabbit, or black-tailed jackrabbit, or you might call it tan-hide rabbit or something. It can have many different common names, but it has one scientific name, Lepus californicus. And the genus is Lepus. Lepus, a lot of rabbits are in the genus Lepus, but the species is specific. There is only one species that goes by the binomial scientific name, Lepus californiensis. There are many other species, like we're Homo sapiens, wolves are Canis lupus, coyotes Canis latrans, and sometimes we'll have subspecies such as Lepus californicus deserticola. Now, if we're going to zoom out on the classification beyond just a single species, broader classification, we have subspecies, then we have more inclusive species, then even more inclusive, the genus is Lepus, when the family Leporidae in the order Lagomorpha, inside the class Mammalia, inside the phylum Chordata, inside the kingdom Animalia. And a lot of mammal study is focused on the ordinal level, and that is going forward, once we get to the living mammals, how we're going to really divide up this course. We're going to think about living mammals in terms of we're going to go order by order. And so there are a lot of orders of mammals, and here's a rough tree based on molecular systematics that shows how they're divided. So first of all, we do have the monotreme and the marsupials, and so there's monotremata, marsupialia, and these are clades. These are groups. All the monotremes are more closely related to each other than they are to anything else in here. The marsupials. Everything else in here is a placental mammal, and we can roughly divide placental mammals into 4 groups. We have the xenarthrans, which are restricted to South America. We have the afrotherians, which they're in Africa, and these are things like elephants, aardvarks, elephant shrews, tenrecs, golden moles, hyraxes, manatees. And some xenarthrans you might know are armadillo, anteaters, and sloths. Then another group is Euarchontoglires. This is our group. This includes primates and rodents plus rabbits. then all other mammals are in this group Laurasia. And this is, well, everything else. This is moles, bats, carnivorans, horses, camels, whales, a whole host of other forms. So these are the main bins that you need to know, like when we go through the different orders of mammals, you need to know, okay, so for example, a dog. Okay, you're going to learn a dog is in the order Carnivora, which is in the grand order or the superorder Laurasia. And so how are all these things related to each other? There's a little bit of a debate here. Different genes and different types of analyses give you different answers, but everybody agrees on these four groups. They agree on Laurasia, Euocontoglires, Afrotheria, and Xenarthra, and also this clade Boroeutheria, that is Laurasia plus Euocontoglires, that is supported by a bunch of things. So we know that Euocontoglires and Laurasia are sister taxa. What there is debate about, though, is after that, at the root of the tree, what happens next? Is Afrotheria at the base, or is Xenarthra at the base? And this third tree here, tree 3, Beza bird, no one really gets that. That's just kind of on there for completeness sake. But there is a lot of debate if Afrotheria is at the base of the tree or if Xenarthra is at the base of the tree. And it changes very easily, so I'm not really confident in saying we know the answer. But we do know how things are related to each other way more than we used to in the past. Here you see on the right, this is a tree from the late 80s or early 90s. I think it came out in 88, which shows at one point it was kind of like, well, here's the different orders of mammals, and we don't really know how they're related to each other. We have some things we're really confident in, like the idea of rodents and rabbits. Well, that's a really old idea. We'll put them together, but it's kind of all these question marks here. It used to be really uncertain how different groups of mammals relate to each other, but now we have a much better idea, but we're still not all the way there. What about fossil mammals? The first part of this course is going to be talking a lot about fossil relatives of mammals. If we include fossils, there are hundreds and hundreds of species. There's thousands of genera of fossil mammals. And we're going to talk about a lot of fossil relatives as we go through each individual order, but we will spend time talking about extinct mammals that have no living relatives as well. And we're also going to go way back to the base of Mammalia, really the base of Amniota, and see how we got from very reptile-like forms to very mammal-like forms. Now, one thing we're going to talk about, and I'm going to use a few times, is the idea of crown mammals versus stem mammals, or crown carnivorans versus stem carnivorans, or crown primates. So, a crown group is the members of a clade that are still alive. So here, this crown group, we can think of this like maybe this is placental mammals and marsupials and monotremes, and they're the ones that have made it today. Well, there's all these extinct fossils that are more closely related to mammals than they are to reptiles, but they're extinct, so they didn't make it all the way. So they're not part of the crown group. They're kind of the stem leading up to mammals. And so this, what we say is talking about a stem group. We're talking about these extinct forms are kind of in between the common ancestor of everything and the living group. And if we say the term total group, we're talking about everything. So again, make sure you know these terms. We use them again and again, and so you'll have more practice in them. But a total group is everything. Crown group is the living representatives, and the stem group is the fossil, the extinct ancestors that are not alive today. Now notice though, not everything in the crown group has to have been made it to here. So see here, this branch right here, this is not alive. This has gone extinct. However, it diverged from everything else after the common ancestor of the crown group. For example, horses. Horses are around today. There's lots of fossil horses, though, that aren't around today. They're still considered crown mammals because, well, they have a close relative that's alive today, and they diverge from horses way after horses diverge from the rest of mammals. And we'll talk about this more when we do our systematics lab in class, in laboratory. if we look at this in an example here using this tree, and this is actually a tree my PhD advisor made for one of his publications. So, some terms here. Eutheria is a stem-based group. Eutheria includes a bunch of extinct fossils, whereas Placentelia, the placental mammals that are alive today, that's a crown group. So Placentalia is a crown group, including the living placental mammals. But if we want to go back all the way to when their ancestors diverged away from marsupials, we're including a lot of extinct forms, and this is called Eutheria, and that is a stem-based group, and together this is a total group. Again, Mammalia, when we talk about this term, this is a crown group. Mammalia is monotremes plus marsupials plus placentals. And Eutheria is another term for placental, and Metatherian is another term for marsupial. Monotremes is monotremes. We don't get really fancy there. So this is a crown group, and even here, these things like multituberculates or eutrichonogonids or other things that are a little hard to say, splachotheroids, these are all part of the crown group, even though they're not alive today, because they diverged after we had monotremes split off from therians. Things like here, docodonts, morganucodonts, tritylodontids, and haramaids, these are stem. These are stem to Mammalia because they diverged before we had monotremes diverge out. And if monotremes had become extinct before the present day, the crown group would be here instead. This is just things that were around kind of when we consider the advent of modern science. Now here we have this, this is considered one of the earliest mammally type forms. If you saw this, you would probably call it a mammal. Well, it's not actually a mammal because this is a reconstruction of Morganucodont, and see, Morganucodont is all the way out here, so we would consider, these are stem mammalia forms or mammalia morphs. They're not actually mammals because Mammalia is a crown group. These are closer relatives of mammals. Another term that I've used a few times is clade. And we're going to start using another term called grade. So grade versus clade. So Mammalia is a clade. We have a clade of mammals, but we can also talk about things being kind of like a grade. What do I mean by that? So here we can say, okay, we have a mammal and we have a reptile. These are clade definitions. So the clade Mammalia, the clade Reptilia. Now here, what about this? Well, we haven't talked about this yet. You probably don't know what this is, but this is a pelycosaur. This is a basal synapsid in the pelycosaur here, and we have, where does this go? Well, if we look at this, if we showed any grade schooler this, they'd say, that looks like a reptile. That looks like a lizard. So even though this is related to the clade Mammalia, we say, well, this is kind of reptilian grade, because its general gestalt, its general collection of features, it looks very what we consider reptilian. It's not in the clade Reptilia, though. It's in the reptilian grade. So again, clade is kind of a group. This is relationship based. So this is the clade synapsida. Based on experimental evidence, based on a lot of detailed observations, we say, like, no, this is on its way to mammals. This is more closely related to mammals than it is to reptiles. So it's closely related to the clade Mammalia, but it's a reptile grade relative of mammals. So we're going to use these terms repeatedly for the next few lectures as we kind of go from very, very reptile-like to very, very mammal-like. And again, the way I think about it is clade. Well, that seems like a science term, so that's based on relationships. Grade, well, what would a grade schooler think of something? That is all I have for the first lecture. What we're going to start talking about in the next few lectures that'll go up this week and next week is we're going to start talking about how we got from this reptile grade pelycosaur, what a pelycosaur is, all the way to mammals. And we're going to go, as you imagine, this took a really long time. It took millions and millions of years, and we're going to make our way through the kind of geologic timescale. We're going to talk about kind of the highlights of how long did this happen, how long did each step take, and just move our way through the fossil record. And so I see everybody on Tuesday.